Biochemistry. Lipid metabolism is a series of biochemical processes involving the processing, storage, and use of lipids in the body. Lipids, which include fats, phospholipids, and cholesterol, play an important role in energy storage, structural components of cell membranes, thermal insulation, and hormone regulation. Lipid metabolism involves various pathways that occur in the body to convert lipids into energy or store them in reserve. Here is a detailed explanation of lipid metabolism. Lipid digestion and absorption. Lipids in food, especially triglycerides, molecules consisting of 1 glycerol and 3 fatty acids, are digested in the digestive tract, namely, a fat digestion. Namely, fat that enters the body will be digested in the small intestine. In the stomach, the lipase enzyme in gastric fluid begins to digest triglycerides into fatty acids and monoacyl glycerols. In the small intestine, pancreatic lipase produced by the pancreas will continue the breakdown of triglycerides into free fatty acids and glycerol. Emulsion and absorption, namely, fatty acids and monoacyl glycerols form micelles, microscopic structures that help absorption by intestinal cells. After absorption, they are resynthesized into triglycerides in intestinal epithelial cells, combined with proteins to form chylomicrons, which are then released into the lymphatic system and eventually enter the bloodstream. 2. Lipid transport in the blood. Digested and absorbed lipids, especially triglycerides, need to be transported to various body tissues to be used as energy or stored. In the blood, lipids are transported by lipoproteins, which are complexes of lipids and proteins. The main lipoproteins are a chylomicron, namely, lipoprotein that transports triglycerides from the intestines to peripheral tissues, such as muscle and adipose tissue, to be used as energy or stored. VLDL, very low-density lipoprotein, namely, lipoprotein made in the liver to transport triglycerides synthesized in the liver to peripheral tissues. LDL, low-density lipoprotein, namely, lipoprotein that transports cholesterol to tissues, especially to body cells for use in cell membranes and steroid hormone biosynthesis. HDL, high-density lipoprotein, namely, lipoprotein that transports cholesterol from body tissues back to the liver to be excreted or reused. 3. Triglyceride metabolism. After reaching the target tissue, triglycerides can be used to produce energy or stored in adipose tissue. Through several processes, including lipolysis. Lipolysis is the process of breaking down triglycerides into free fatty acids and glycerol. This process occurs in adipose cells, fat cells, when the body needs energy. The enzyme hormone lipase, HSL. Hormone-sensitive lipase plays an important role in the breakdown of triglycerides. Lipolysis is regulated by hormones such as 1. Glucagon and epinephrine, adrenaline, which increase lipolysis to provide free fatty acids as an energy source. 2. Insulin, which inhibits lipolysis, helps store lipids in the form of triglycerides. B. Beta oxidation of fatty acids. Free fatty acids released by lipolysis then enter target cells, especially muscle and liver cells, and undergo beta oxidation in the mitochondria. Beta oxidation is the process of breaking down fatty acids into 2 carbons per molecule, acetyl-CoA, which is then used in the citric acid cycle, Krebs cycle, to produce ATP, which is the main source of energy for cells. This process involves several steps. 1. Fatty acid activation, namely, fatty acids are converted into acyl-CoA with the help of the enzyme acyl-CoA synthetase and ATP. 2. Transport to mitochondria, namely, acyl-CoA is transported into the mitochondria by the carnitine transport system. 3. Beta oxidation, namely, acyl-CoA is processed in 4 steps to break it down into acetyl-CoA molecules. Each round of beta oxidation produces NADH, FADH2, and acetyl-CoA, which can then be used to produce ATP through the electron transport chain and oxidative phosphorylation. IV. Fatty acid biosynthesis, lipogenesis. Lipogenesis is the process of forming fatty acids from acetyl-CoA. This process occurs mainly in the liver and adipose tissue, especially after eating foods rich in carbohydrates. When glucose is available in excess, glycolysis produces pyruvate, which is then converted to acetyl-CoA. This acetyl-CoA is used to form fatty acids through several steps. A formation of malonyl-CoA, namely, acetyl-CoA is converted to malonyl-CoA through the enzyme acetyl-CoA carboxylase. B. Formation of fatty acids, namely, malonyl-CoA is combined with acetyl-CoA to form a long carbon chain that forms fatty acids. This process involves the enzyme fatty acid synthase, which adds 2 carbons to the fatty acid chain each cycle. The newly synthesized fatty acids can then be converted into triglycerides for storage in adipose tissue or transported to other tissues using lipoproteins such as VLDL. Versus cholesterol synthesis. Cholesterol is an important lipid used to form cell membranes, steroid hormones, such as estrogen, testosterone, and cortisol, and bile acids. Cholesterol synthesis occurs mainly in the liver and involves several stages. 1. Formation of mevalonate, namely, acetyl-CoA is converted to mevalonate through a series of reactions catalyzed by the enzyme HMG-CoA reductase, which is the main regulatory point in cholesterol biosynthesis. 2. Cholesterol formation. Namely, mevalonate is then converted into isoprenoids and finally into cholesterol through several synthesis steps. The synthesized cholesterol can be used in cell membranes, secreted in bile, or used to form steroid hormones. V. Lipid storage and regulation. Lipids are stored in the form of triglycerides in adipose tissue, fat as energy reserves. Triglyceride storage is carried out through the process of glycogenesis, which is triggered by the hormone insulin after consuming energy-rich foods. When the body needs energy, for example, during fasting or physical activity, the hormones glucagon and epinephrine stimulate lipolysis, releasing free fatty acids that can be used to produce energy through beta oxidation. 7. Functions of lipids in the body. Lipids have various important functions in the body, including an energy source, namely, lipids, especially triglycerides, are an efficient energy source, providing more energy per gram compared to carbohydrates and proteins. B. Formation of cell membranes, namely, phospholipids are the main components of cell membranes, providing structure and stability. C. Thermal insulation and protection, namely, body fat helps insulate the body against extreme temperatures and protects vital organs from physical impact. Hormone precursors, namely, cholesterol is used to form steroid hormones such as cortisol, estrogen, progesterone, and testosterone. Energy storage, namely, lipids are stored in adipose tissue to be used as energy reserves when needed. 8. Regulation of lipid metabolism. The lipid metabolism process is regulated by several hormones that work together to maintain the body's energy balance, including insulin, namely, activates lipogenesis and inhibits lipolysis. Insulin regulates energy storage in the form of triglycerides. B. Glucagon and epinephrine, namely, increase lipolysis and provide free fatty acids for use as energy. C. Thyroid hormone, namely, affects lipid metabolism by increasing the rate of fatty acid oxidation. Conclusion: lipid metabolism is a complex and very important process in the body for energy regulation, storage, and the formation of structural components and hormones. Lipids obtained from food are digested, absorbed, and transported to body tissues for use or storage. While lipid metabolism that occurs in cells converts fatty acids into energy used for cellular function. These processes are tightly regulated by hormones to maintain the body's energy balance.